Okay, for exam Friday, make sure that you pace yourself appropriately because we've got another class in here at 10 o'clock, and so you do need to be finished by 9:55. And so at 10 till, I'll start handing out exams, so get over here as quickly as you can to get going. Same length of the exam as I mentioned last time, same number of questions, same kind of general breakdown of questions, so you should know what you're up against. I'll put it on the exam as well for a reminder, but there will be questions asking you about where does this happen, where does this happen. Remember I had that PowerPoint slide that said this is some really basic stuff you should know about cellular respiration. So when you're answering those questions, please make sure you remember that we're considering that transition reaction to be part of what happens in the citric acid cycle, even though technically it's not. But I didn't want to break it out and have another option of transition cycle. So anything from pyruvate on through the citric acid cycle is considered to be part of the citric acid cycle. Okay? And when you see the exam, don't freak out because there will be some questions about, where is ATP produced by a substrate level phosphorylation? And you'll have a lot of choices. There'll be glycolysis. There'll be citric acid cycle. There'll be electron transport system. There'll be glycolysis and citric acid cycle. There'll be glycolysis and electron transport. So all the combinations will be shown. So for those questions, it's like choices A through H, okay? Otherwise, it's, the normal 4 or 5 choices. But for those, you have to know if it happens in more than one of those cycles. You have to know that information. Okay? So let's finish up exam material for Friday today. And we were in the midst of talking about the electron transport chain. Remember that the purpose of this is really to reoxidize these coenzymes. That's what it's there for. A bonus of how it does that is that it can make ATP by chemiosmosis. So what pathway is this recycling mechanism kind of similar to that we've already seen? What's kind of this analogous to? What other pathway do we have its intention is to recycle those coenzymes? Glycolysis? Recycle coenzymes in glycolysis? No. Well, what's got to accompany glycolysis? Either this recycling mechanism. What's the other way we can recycle coenzymes in accompaniment with glycolysis?
Fermentation.
The final steps of fermentation, okay? So when we convert pyruvate into ethanol, that is the recycling process that's associated with glycolysis to keep it going. And so those final steps are kind of analogous to this electron transport chain. But what does this do that those final steps of fermentation don't do? Creates a lot of ATP, okay? That's where you get your big payoff. Okay. This is the matrix of the mitochondria. This is this intermembrane space in our mitochondrial model out here. So matrix here, intermembrane space here, membrane here. And as these particular reduced coenzymes are oxidized, protons get pumped out to this intermembrane space. Now, I have a real issue that Dr. Morton pointed out to me with this slide in that it is not entirely accurate. What they really should be doing is putting a 2 in front of each of these H pluses, because for every one of these coenzymes that donates its electrons to the electron transport chain, it pumps out 2 H pluses here, another 2 here, another 2 here. And then as 2 of those flow through the channel, that gives you enough energy to make ATP. Okay? So if you've got your slides with you, you might want to make that notation or change. And if not, I would make a little notation in your notes and make that change on your slide. So this should be 2, 2, 2, 2, 2, and then we make 1 ATP out of that. If you don't understand that, when we start adding up ATPs, it's going to not seem to make sense. High concentration, low concentration. High free energy, low free energy. Natural tendency of those protons is going to be to move from here to here. Without this ATPase channel protein, will that happen? Well, those protons go from here to here. Why not, Jesse? Why not?
Are they charged?
They're charged, yeah, protons. This hydrophobic core of the membrane is not going to want to let those charged molecules across. So this channel protein facilitates their movement from high free energy to low free energy. And basically the way you can envision this is that ATPase channel protein is not just a channel, but it's a little kind of rotary generator, if you like, that as two protons flow through here, it generates the energy to phosphorylate an ADP to make ATP. Okay? So the electron transport chain basically charges up the battery, if you like, by creating high free energy out here, and then that free energy is tapped or used during chemiosmosis to drive the endergonic synthesis of ATP. So ATP is up here, ADP and phosphate are down here. If we want to go from ADP and phosphate back up to ATP, that requires an input of energy. The source of that energy is a hydrogen ion gradient. The gradient's generated during the recycling of coenzymes that occurs during the electron transport system. And that process will only continue if there's a readily available supply of oxygen, because we need some place for those electrons to finally go. Otherwise, this system backs up. These can't be oxidized. And so what is the cell forced to do? The cell's forced to oxidize these through fermentative pathways, which don't generate all of that additional ATP. Now, the one remaining piece of the puzzle that I've talked about a little bit already that I want to make sure you can appreciate is, why does this electron transport chain result in hydrogen ions being pushed or pumped from in here to out here? What is it about that electron transport chain operating that accomplishes that crucial, essential goal? Okay, this one's a little bit hard to see. Let's see if I can magnify this a little bit. Yeah. Okay, this little hand-drawn diagram, which is pretty crude obviously, this would represent the cytoplasm of the cell. This represents the outer membrane of the mitochondria. Here is our intermembrane space right here. Here's our mitochondrial inner membrane, and here's the matrix. Okay? So same setup we saw on the slide before, just not as pretty. Okay? As a result of recycling these coenzymes, we are pumping hydrogen ions, and the numbers here are correct, out into this intermembrane space at the expense of the matrix. Okay? So the net movement is going to be protons moving from here out to here. And then as those flow through the ATPase channel protein, for every 2 of those that flow out, that will give you an ATP. Okay? So remember our equivalencies. What did we say? We said a reduced NAD gave you the potential to form how many ATP? 3, right? Why? Because when a reduced NAD donates to the electron transport chain, it pumps out 2 H pluses here, another 2 here, another 2 here. Those 6 total going through the ATPase channel can give you 3 ATP. Okay? Reduced FAD donates to a different point in the electron transport chain, a little bit more downhill. When it donates, it pumps 4 hydrogen ions out here. When those flow through the ATPase channel protein, that's only going to give us 2 ATP. Okay? So remember that equivalency. A reduced NAD gives you 3 ATP. A reduced FAD gives you 2. It has to do with how many hydrogen ions are pumped out here during the electron transport chain. Okay. 3 pieces of crucial information you need to understand to appreciate why this is happening. Number 1, in the matrix of the mitochondria there is water. And water will be spontaneously dissociated into protons and hydroxide groups. So this will add basically protons to this pool right here. Okay? Number 2, not all of these electron transport compounds are the same. Some of them accept hydrogens when they become reduced. And what's a hydrogen made up of? Proton and an electron. Some of the compounds will only accept the electron portion of the hydrogen. So what's going to be left unaccounted for? A proton, right? Somebody's trying to give you a hydrogen and you say, sorry, I'm only taking the electron. There's a proton left there. Okay? And these compounds tend to alternate in the electron transport chain. Important point number 3. These are asymmetrically distributed. They're unequally distributed. Some tend to face this side of the membrane, closer to the matrix. Some tend to face this side of the membrane, closer to this intermembrane space. So, look what's happening here. Reduced NAD, produced during citric acid cycle, is going to be recycled. It's going to dump its hydrogens to this first compound in the electron transport chain. Previously, this was oxidized. When it accepts those 2 hydrogens, it's now reduced. What does it want to do? It wants to give up its hydrogens to this compound so it can accept more again. Keep that bucket brigade going. Keep handing your bucket off so you can take another one. But this compound is an electron accepting compound only. It doesn't want those hydrogens, it just wants the electrons. So it grabs the electrons. Now there's 2 protons that are unaccounted for. Are they going to be happy sitting inside of that hydrophobic core of the membrane? No. And because these compounds tend to face this inner membrane, what's more likely? That those protons get spit out here, or that they're going to go all the way across back here into the matrix? They're more likely going to get spit out here. Okay? This compound now has got a couple of electrons. It wants to get rid of them so it can accept more over here. So it grabs electrons. But this compound is a hydrogen-accepting compound. It takes the electrons, but it needs some protons as well to completely reduce it. Where does it get those protons? From the matrix, from the dissociation of water. So now it's effectively got hydrogens. They're transported over to this compound. It'll take the electrons, not the protons. It donates electrons here. This needs some protons to be reduced. It's getting its electrons, it needs some protons. From where? From water. And that continues until the electrons are finally passed off to oxygen. To reduce oxygen to water, you not only need electrons, you need protons. So we are depleting the H pluses here. They are being accumulated out here, and that gives us that hydrogen ion gradient that we use to make ATP. This is where you get most of the payoff of aerobic cellular respiration. Now, this might be a little confusing to you. Just a second, I will point out this inconsistency that seems to be here and come back to it, and I'll explain why this is the case. Okay, so let's go back to kind of big picture. Okay, so here's our glycolysis, here's our citric acid cycle, and here is our electron transport chain. Okay. Glycolysis, remember, produces 2 ATP by substrate level phosphorylation. Citric acid produces another 2 by substrate level phosphorylation. And again, this is per glucose. So there's 4 already. Okay? Glycolysis produces 2 reduced NADs. If this is going to keep going, these have got to be recycled. If there's oxygen present, then those reduced NADs can get into the mitochondria and run through the electron transport chain. Now here's where you've got your little bit of confusion. This membrane is not particularly permeable to reduced NAD. The mitochondrial membrane isn't. This is happening in the cytoplasm. And this whole big reduced molecule can't get across that membrane very easily. So there's a little exchange system that's going on. Oxidized NAD in both, or FAD, both in the matrix of the mitochondria, can grab those hydrogens from this reduced NAD. So this donates to these. That recycles this. Okay? Reduced NAD or reduced FAD can potentially pick up those electrons. So this can shuttle to this or to this. When these go through the electron transport chain, this gives you 3 ATP per reduced NAD. This only gives you 2. And so that's why it says about 36 or 38. It's because of this little wrinkle right here of this exchange that happens between this and these. Ready?
Can you explain that again? I didn't understand.
Okay. So these are being produced in the cytoplasm, right? From glycolysis. These, to be recycled, you would think would have to get into the mitochondria, right? Go through electron transport. But the membrane's really not permeable to these. So what happens? These move up to the membrane, and inside there's some oxidized NAD or FAD that can grab the hydrogens from this. So now this is recycled. It's equally likely that this can grab the electrons from this or this can. Both can happen. When these then go through the electron transport chain, they give you different amounts of ATP. Remember, this gives you 2 per reduced FAD. This gives you 3. And so that is why we have a potential discrepancy between, you might get 36, you might get 38. It depends upon which one of these picks up those electrons from this.
So then are the NAD and FAD that are oxidized by the electron transport chain, are they the ones used in the mitochondria?
Yep.
Then, okay.
Yep. So don't get too hung up on this. I'm fine with you, for practical purposes, saying 38's the number. In fact, that's the number I've been telling you. And that basically kind of implies that this goes into the mitochondria and runs through the electron transport chain. In reality, it's not quite that simple. And so I'm pointing this out to you because you will see this in your textbook if you start reading through your textbook to study. And I don't want you to get hung up on this little kind of minor little wrinkle when you're doing kind of your accounting of where ATP comes from. So I would basically say 38 ATP per glucose, and then just kind of assume that really this can get into the mitochondria to be recycled by the electron transport chain. Sometimes it's easier to simplify things so you don't lose track of the big picture. So this is just another way to kind of show you where the energy payoff comes from. Okay, so here's where all of your reduced NADs come from. 2 from glycolysis, 2 from the transition reaction, another 6 from citric acid cycle, another 2 from citric acid cycle. And remember, these are numbers per glucose. And then this will give you 4 in electron transport, 2 times 2. This will give you 18, 6 times 3. This will give you 6, 2 times 3. And then here you've got this 4 or 6 dilemma. And so that gives you your total from oxidative phosphorylation of 32/34. Let's say 34. Okay. And then if you add the other 4 from substrate level phosphorylation, because this is all oxidative, then you get your total of 38. The big thing that I hope you appreciate and understand from this is where you get your big energy payoff. And this all depends upon a functioning electron transport chain. And without oxygen, this doesn't function. You don't get this payoff. And really, all you do during glycolysis is add a couple of steps to recycle this back to NAD, and then you get your 2 ATP. That's all you get out of it. A couple more slides, just, oops. Couple more slides, just give you some summaries. And so this is just kind of a written summary of what happens in fermentation, what happens in glycolysis, what happens in citric acid cycle or Krebs cycle here, and then what happens in the electron transport chain. So this would be a good summary to study from. And then this is an overall summary of cell respiration. This is cell respiration in a big long paragraph. And so you might want to make sure that you look through that slide and read through that, and that this makes sense to you, because this is really just boiled down into the very key elements. If all the accounting really just blows your mind, don't spend an excessive amount of time trying to master all of that at the expense of other material. There will be a couple of questions that test your ability to kind of account, but that's not going to be a major set of questions on the exam. The big thing that you need to concentrate on are the big picture items, like those questions that I gave you at the beginning, saying, where is ATP produced? Is CO2 produced here? Is NADH reduced here? Etc. Those are the big picture things that will get you the bulk of the points. Some of these kind of more a little bit detailed questions, might be the difference between getting an A and a D on the exam, but certainly don't just spend all of your time on that particular information. Okay. Another important point about cellular respiration, and I can't stress this enough. We have up till now been stressing the catabolic role of cellular respiration. Remember what catabolism refers to. Catabolism involves taking large complex molecules like glucose and breaking them down through a set of reactions to form simpler molecules, carbon dioxide and water, releasing energy, hopefully harvested in the form of ATP, and that this overall is a spontaneous reaction which involves the oxidation of this molecule to form CO2 and water. This is what you do predominantly when you have a high energy demand in your cells and you need a lot of ATP. So muscle cells that are very active are going to be doing this a lot, breaking sugar down to make ATP. But cell respiration has another role in metabolism. Cell respiration also has an anabolic role in metabolism. Remember what anabolism is. Taking simpler molecules and making more complex molecules out of them. That's an energy requiring process. Typically, those molecules become more reduced during the anabolic reactions. It's not spontaneous. There are mechanisms whereby the body can use a number of different food sources in catabolic cellular respiration. Okay, glucose is obviously the most evident one. If you eat some glucose, this is what's going to happen, and you're going to burn it up to make ATP potentially. What if you consume other sugars, like sucrose? Well, if you have the enzymes that can convert sucrose into glucose and fructose, sucrase enzyme, then the glucose can run through cellular respiration. The fructose can be chemically modified so it can go through there as well. What if you've got starch? Well, if you have the enzyme amylase, then you can degrade starch into sugars, and then glucose will run through cellular respiration. But are carbohydrates the only source of energy? No. What about fats? Fats are a source of energy. If you have the molecules that the enzymes that can break fat down into their glycerol and fatty acids, then there are other metabolic pathways that can convert glycerol into this compound, glyceraldehyde 3-phosphate. That's a part of glycolysis. So basically, glycerol can feed into glycolysis at that point, then make pyruvate, okay, and work as normal. What about fatty acids? Fatty acids can be converted into acetyl-CoA. So they can feed into cell respiration at this point. What about proteins? Can you derive energy from proteins? Sure. What do you have to do? You have to break those proteins down first into their individual amino acids. Well, we have digestive enzymes in our stomach and our gut that can help us do that. Amino acids can then be deaminated. Deaminated means that the amino group is removed. Now, remember what an amino acid is. An amino acid has a central carbon, a hydrogen here. What functional group is this? Carboxyl. What's this? Amino. And then this R group varies depending upon which of the 20 amino acids you're talking about. It could be as simple as a hydrogen in glycine, or it could be a complex ring structure in tryptophan. Okay? If we're going to use these as energy, we've got to remove this amino group first. And then the rest of the molecule can be converted into, depending upon the amino acid, maybe acetyl-CoA. Or maybe one of these compounds in the citric acid cycle. But we've got to lop off the amino group first. During metabolism, ammonia starts to accumulate in cells. So what do? Cells move it out into the blood. You know why they do that? Why do cells get rid of ammonia that they generate during metabolism? It's bad. It's toxic. Okay? Yep. And then in the circulatory system, ammonia is moved to the excretory organs, like the kidneys, and converted into urea. And then we void that ammonia. Okay? But the ammonia is a byproduct of utilizing proteins as an energy source. After we lop off the amino group, then the remaining part of the molecule can feed into glycolysis here, or here, or here, depending upon what amino acid's being metabolized. Okay? So cell respiration is more about than just getting energy out of glucose. All food molecules can potentially feed into the glycolytic or Krebs cycle pathway, citric acid cycle pathway, to generate energy. But still, that's all catabolism. What about anabolism? Could I potentially pull glyceraldehyde 3-phosphate out of this pathway and use it to make glycerol? I could. The cell could. Could the cell potentially pull acetyl-CoA out of this pathway and instead of using it for the citric acid cycle, go through a series of reactions to make fatty acids? Yep. And then once you've got glycerol and fatty acids, if you have the proper enzymes, can you make fats? Yeah. When would you potentially do that?
Like if your cell membranes are damaged, then you can make more fatty acids.
Okay, yeah, I mean, if you need to replace membranes, for example, you would want to do that. Would you probably be doing this, making fat, if the cells were really active and needed a lot of ATP? No, that would favor complete catabolic breakdown. But what if your cells aren't really active? What if you've consumed a lot of sugar and then you don't do anything for a long time? Well, part of that sugar could be broken down to make ATP, but some of it could be diverted into synthesis of other compounds like fats. So the idea that sugar can turn into fat, particularly in cells that aren't very active, is very consistent with this whole metabolic system. Can you make proteins out of these compounds like sugar? Potentially. Pyruvate could be converted into amino acids, but you need a source of nitrogen to do that. Humans don't do this, really. But plants can. This is one of the reasons why you've got to supply a lot of nitrogen in the soil to plants. Because they make this by what process? Photosynthesis, okay? So they can make their sugars. But can plants make proteins out of sugars? Yeah, if you give them the right amount of nitrogen. And if they've got the other enzymes that can then convert the sugar into molecules that can be converted into amino acids. So it's really important to understand that cellular respiration, glycolysis, citric acid cycle, have a role both in the catabolism of metabolism, but also in the anabolism of metabolism. Okay? They play a role in both energy production and providing what people usually refer to as carbon skeletons that can then be converted into other molecules for synthesis. So do cells have kind of a demanding, conflicting demand about what they do with things like sugar? Yeah. If there's a lot of energy demand, then this will be favored. If there's not a lot of ATP demand, they'll still do some of this, but then this might be favored as well. Okay? So the cell is constantly going through that balancing act. And one of the things that helps cells to do that is regulation by feedback inhibition. Some of these enzymes are very sensitive to being temporarily shut off by other molecules. ATP can feed back and temporarily inhibit this. AMP, which is a breakdown of ATP hydrolysis, can stimulate this. So if there's a lot of ATP already, this tends to be inhibited. If there's not much ATP, this tends to be stimulated. So that idea of feedback inhibition is really important in regulating the cell's kind of conflicting demands for what it does with glucose, for example, at any point in time. Does it break it down, or does it convert it into something else?
How do you get AMP?
Don't worry about that right now. If you want to ask me later, you can. It's just a product of breakdown of ATP. Okay. Okay. That's cell respiration. Anything that's just really confusing you still? All right.
Can we go back to that previous, the following slide, please? The protein, fat, carbohydrate, metabolism. Yeah. I think it's useful to note if you are dieting and you say you're on a low carb diet And so you're not making any ATP by that dual pathway. Your body's going to burn a lot of fat in order to make the ATP. I mean, that's part of the process of dieting. But if you do this in extreme, if you're bulimic or, what's the other term? Anorexic. And you're not getting the carbohydrates and the fats, where is your body going to get its ATP? It's going to start digesting your proteins. And this is one of the pathologies associated with those two disorders. Is that when you do not get enough calories from carbohydrate and fat, your body basically starts digesting itself. And that's why so many individuals who are anorexic or bulimic, their body starts digesting their muscles. And it all
That's not good.
Yeah, that's not good.
Another interesting thing, years ago, some researchers developed what they thought was a great diet drug. And the way the diet drug worked was it uncoupled electron transport from generating this hydrogen ion gradient. So electron transport was still occurring. But if the hydrogen ion gradient wasn't being formed, then you really weren't making ATP. And the idea is that people would be burning energy, but then not producing a lot of this to then make fat. This particular drug ended up having to be pulled from the market because people were dying because they weren't producing enough ATP. But this molecule that could kind of separate or uncouple electron transport from the hydrogen ion gradient was at one time thought to be a real kind of miracle diet drug. Didn't work out very well. Got time for a couple of questions. Don't worry about clickers. So what happens if we deprive a cell of oxygen? Five will happen, okay? ATP is not going to be produced by oxidative phosphorylation because there's nothing to accept those electrons, so the electron transport system backs up. We can't recycle reduced FAD if the electron transport chain is all backed up. Therefore, the citric acid cycle will stop. Why? Because there won't be oxidized FAD present or oxidized NAD present. I just feel like you're out there giving me gang signs. This is the false one, right? Where is it? Inner membrane of the mitochondria. There indeed are two types of carriers, the hydrogen and electrons. Cytochromes are iron-containing proteins, and those compounds are indeed asymmetrically distributed within the membrane. Remember your equivalency in the electron transport chain, 1 reduced NAD, 3 ATP. Correct answer is 12. If the question was FADs instead, the answer would be 8. What about the mitochondrial inner membrane? All of those are correct. That membrane's normally impermeable to H+, except that it's got a channel protein that can let them through. which is also an ATPase, separates those two compartments, and it's where the electron transport chain is located. Okay, if you are comfortable with aerobic cellular respiration, and especially the chemiosmosis that we just talked about in the mitochondria, your life will be a whole lot easier when we tackle photosynthesis next. Because photosynthesis depends upon some of the same principles. In photosynthesis, we do produce ATP in the chloroplast by chemiosmosis. What's the big difference? What is the source of energy that's used to make that hydrogen ion gradient? It's not the oxidation of glucose. It is the photons of light that ultimately are the energy source to make that hydrogen ion gradient to make ATP by chemiosmosis in photosynthesis. So if you understand this, photosynthesis will be much easier. Couple of quick comments. My goal by the end of the semester for end of the chapter quizzes is that no one will miss a question that was one of the ones at the end of the chapter. Pretty much regularly, the first two questions on your quiz have come from the end of the chapter, and people are still missing those. And then read questions carefully on quizzes or exams. How many ATP are produced by citric acid cycle by substrate level phosphorylation for glucose? 2, right? Any produced by oxidative phosphorylation? What percentage of the ATP produced in the citric acid cycle is produced by substrate level phosphorylation? All of it, 100%. 2 out of 2. Okay? Some of you missed that question on your quiz because you didn't read the question carefully. Okay? That's why many of you going 4 out of 38. The question asked how many in the citric acid cycle. So please make sure you read the questions carefully. Okay? Tomorrow we will start photosynthesis, and that won't be on the exam obviously. That'll still over the 30 exam. Okay?